I'm calling it the Goldilocks decision. Of species of crabs, cancer crabs, lady crabs, blue crabs, and a variety of crustaceans, including mantis shrimp. Hudis went on to organize several volunteer groups in less than one up in the upper reaches of the Providence River, north of Fields Point, very low concentration around Barrington Beach and the Warren River, and Greenwich Bay, all extending down in a kind of a tongue into the lower West Passage. And that tongue has reached as far south as Quonset and told them to reduce their nitrogen content. And their initial goal was to reduce the nitrogen by 30% by 2005. You can see from that graph that they actually achieved that goal very well. So in 2005, we received a grant from NOAA. Research goal was to do observations and model the low ox to the nitrogen reduction. And my role in that program was to do the field observations to support the modeling effort. So in 2006, we started a bay-wide nutrient standing stock concentrations program, and we measured 12 sites to estimate daytime primary production and nighttime respiration. The main goal of the program, however, was to develop this very innovative model, and the ecological portion of the model was performed by Jim Kramer and Jamie Vaudry at the University of Connecticut and Mark Brush at the Virginia Institute of Marine Science. The goals for my presentation here today are the decrease in standing stock of particularly nitrogen in the bay as a consequence of these regulations. And then I want to examine with you some of the habitat changes associated with the nitrogen reduction. And we'll look at the primary production and respiration rates in the bay. We'll look at the frequency of hypoxia, summertime hypoxia, particularly in the upper bay. The indicator I wanted to look at was fish biomass. So let me introduce you to the team that carries out this work. Katie Copeland is not here with us anymore. She was my master's degree student, and she's now at the University of Maine working on her PhD program. But the three technicians that perform all this work are Edwin Requintina there. And on your right is Laura Reed. She does our nutrient sampling, nutrient analyses, and data management for nutrients. She's also working for Randy right now. The fixed site and buoy network has to be serviced every 2 weeks because of fouling. If you've been out of the bay, you've probably seen these yellow markers as you've been driving around. That's one of our buoys. We have 12 sites in all. And at the buoy, they take a profile. They also measure light through the water column and alkalinity. This is Heather making one of those profiles. And when we bring the sensors up, we have to exchange them every 2 weeks because of fouling. And on your right there, the one from western Greenwich Bay is way more foul than the one from the buoy out in the bay proper. You can compare the fouling here. So look at all those tunicates on the sensor of the West Greenwich sensor compared to the sensors that are still pretty clean for the Quonset Point buoy. So our fixed site and buoy monitoring network are at the location of these red triangles. There's 12 of them on here. And I should probably name them for you because I'm going to keep referring to them as we go along. So the furthest north one up there is Phillipsdale, and then the Providence River, Bullock's Reach, Connecticut Point at the mouth of the Providence River. In Greenwich Bay, we have Greenwich Bay at the western end there, and then Sally Rock in the middle, North Prudence, Mount Hope Bay, Mount View, Papa Squash in the East Passage, Quonset Point, T Wharf, which is at the southern tip of Prudence Island, and this is one of the partners in this network, the Estuarine Research Reserve. And the partner in the Providence River and running the Phillipsdale one is the Narragansett Bay Commission. And then we have one GSO site down here that we're responsible for. But we manage all 12 of the sites, and we manage the data from all the sites. In addition, on this graph, you'll see there's some opaque dots which are numbered. These are the location of our nutrient sampling stations that we sample every 2 weeks in the summertime and monthly during the wintertime period or the colder months. And my student Jason Krumholtz got his PhD on the nutrient distribution in the bay during the 30% nitrogen reduction period from 2005 to 2010. And he's done these wonderful graphics showing the gradient of, first of all, dissolved inorganic nitrogen in yellows and reds there. And you can see that the problem areas are in the Providence River area, the Ohio Ledge area, Greenwich Bay and that tongue coming out into the West Passage, and Mount Hope Bay. It's where you have the higher concentrations. And at the mouth of the bay, the concentrations are less than 3 micromoles. They're also high in the Providence River, the Ohio Ledge area, Greenwich Bay, particularly Mount Hope Bay, and concentrations at the mouth are less than 1 micromole. So the lower part of the bay has always been pretty low in nutrient concentrations. So this is a series of bar graphs that documents the changes in nutrient concentrations, total nitrogen in this case, versus this north-south gradient from Phillipsdale, the Seekonk River, down to Rhode Island Sound. And we did several surveys, one way prior to the nitrogen reduction in 1998. That's the dark blue bar, the first dark blue bar. And then I averaged the 5-year period of 2006 to 10 when we had the 30% nitrogen reduction. And then the individual years 2012, 13, 14 to give you a sense of the interannual variability and also to show you what the 50% nitrogen looked like in 2013, 2014. So if we choose one of these to look at, say Connecticut Point at the mouth of the Providence River, we went from almost 80 micromole of total nitrogen down to less than 40 micromole with the 30% reduction, and then you can see some down and up activity here. And then the final blue bar is 2014 when we had the 50% nutrient reduction. You can see some of these stations don't fit the gradient very well. Greenwich Bay leaps up. We still have a big nutrient problem in Greenwich Bay. We still have a big hypoxia problem in Greenwich Bay. And then dissolved inorganic nitrogen, this is the same sort of graphic, done the same way. And we did a survey in 1980, long before nutrient reductions, in the first dark blue bar. And we can look at that at North Prudence, where it's about 10 prior to nutrient reduction, goes down to less than 10 with nutrient reduction, not as much as one might hope. But with 50% reduction, it's more like about 5 micromole. Again, you'll notice along this gradient that Mount Hope Bay, Greenwich Bay, which have other sources of nutrients, don't fit this gradient so well. So I had to statistically test these reductions. So I took the 3 periods of prior to nutrient reduction for total nitrogen and dissolved inorganic nitrogen, the top graph and the lower graph, and then that period of the 30% nutrient reduction from 2006 to 10, and then the period of the 50% reduction, 2013 and 2014. I plotted the log of the total nitrogen, the log of the DIN, and then I tested these lines, and in both cases, there's a statistically significant decrease from prior nutrient reduction to the 30% reduction, and then from the 30% reduction to the 50% reduction. So the reduction is, variable, but statistically significant. So what does this look like in percentages? How well did Rhode Island DEM meet their goals? And actually they did better than meet their goals because, it helped that a lot of wastewater treatment facilities in the watersheds were also employing the same process at the same time and reducing their nitrogen input to the rivers that feed into the bay. So in fact, they achieved more than their nutrient reduction of 50% goal. And the top table here is for dissolved inorganic nitrogen from the northmost station in the bay, Fields Point in the Providence River, down to beaver tail, and then total nitrogen along that same gradient. So the average decrease for dissolved inorganic nitrogen is 60%, and for total nitrogen is 58%. Pretty good, I would say. The phosphorus is a different story. That didn't happen because of recent changes in wastewater treatment facility. It happened because it was realized in the late 20th century that phosphorus was a source of eutrophication in freshwater lakes primarily. And in consequence, the companies started to take phosphate out of their detergents. And so by the 1990s, really, the phosphate input to the bay had been decreased. But we can look at what the size of that decrease was because we had some surveys prior to that decrease as well. And again, this is the north-south gradient of stations in the bay for dissolved inorganic phosphate and total phosphate. So the total dissolved inorganic phosphate decrease was on average 61%, and the average decrease for total phosphate was about 54%. Very equivalent to the nitrogen decrease. So Heidi Travers at Rhode Island DEM sent me this graph the other day. It's based on different data than what I have used, and it's a nice verification of the same data for standing stock concentrations. And she's showing that the wastewater treatment plant input from the 11 plants directly going into the bay is after 2005 they achieved about, by 2011 they achieved a 27% decrease overall, and in 2013 they achieved actually a 62% decrease. This is the amount of nitrogen going into the bay from the wastewater treatment plants. So we have nice verification from 2 different data sets that the decrease was what we have been claiming it to be. Okay, let's look at some of the habitat indicators. I always like to bring in the Merle tanks, so I'm going to talk about one of our early experiments here, where the x-axis is an enrichment multiplier, where 1x is the nitrogen input, the average nitrogen input from wastewater treatment facilities directly to Narragansett Bay. And the y-axis is primary production in grams oxygen per meter squared per year. What this graph shows is we have kind of a hyperbolic tangent that kind of asymptotes as you get to higher nutrient concentrations. It never levels off. It always continues to go up with respect to primary production, but it goes up slower and slower. So for initial nutrient reductions, going from 32x to 16x, which is a huge decrease in nutrients, you only get about 100 grams of oxygen per meter squared per year decrease. But if you get down into the exponential part of the curve, and you go from 2x to 1x, then you get about 200 grams oxygen per meter squared per year reduction. A bigger bang for the reduction in nitrogen as you get decrease as you get to the exponential portion of the curve. So this is what we thought might be going to happen. And here are our primary production measurements plotted on the positive part of this axis, the y-axis. Surface production in grams oxygen per meter cubed per day. And then the negative portion of the axis is respiration in grams oxygen per meter cubed per day. And I've plotted here again the north-south gradient in stations from Phillipsdale, the Seekonk River, Bullock's Reach, the Providence River, Conimica Point, North Prudence, Mount Hope Bay, Greenwich Bay, Papasquash, Mount View, T-Wharf, Quonset Point, and finally GSO. And the first bars of our study conducted in 2004 prior to the nutrient reduction, we didn't have as many stations back then, are very dark green. And then the 2 most recent stations, 2013 and 2014, are with the 50% nitrogen reduction. A lot of interannual variability. But we can look here at North Prudence, and you can see we started out with about 1 gram oxygen per meter cubed per day, and with the 50% reduction, we're about 0.5. The other thing to notice here is Greenwich Bay. It's still got way too much primary production and way too much respiration. Mount Hope Bay is a little bit high with respect to the gradient as well. And then I plotted this another way along the south to north latitudinal gradient going from Graduate School of Oceanography dock up to Phillipsdale. And the y-axis here is primary production grams oxygen per meter cubed per day. The black triangles are the 6-year period from 2006 to 11, and the red dots are the period of the 50% nitrogen reduction, 2013-2014. Generally, they're less than the black triangles. Not for Phillipsdale. They still have a nutrient problem up there in the Seekonk River. And I haven't yet been able to show that these decreases from the 30% nitrogen reduction to the 50% nitrogen reduction are statistically significant. But it's a very strong trend of decrease in primary production. And this converts it into percentages. This is the decrease in surface system production during the nitrogen reduction. And again, we're going from stations in the Province River, Bullock's Reach, Connecticut Point, down to GSO. And the average decrease prior to nutrient reduction to the 30% reduction is minus 21%. The decrease from the 50% reduction to the 30% reduction to the 50% reduction is minus 17%. And the overall reduction from 2004 to 2013-14 is minus 33%. Those percentages are a little deceptive on average because the middle of the bay has decreased a lot more, minus 44%, minus 45%. And I've got Greenwich Bay in there, which is not decreased all that much. So for the vast part of the bay, the percent decrease has been almost equivalent to the amount of the nitrogen decrease. Not quite, but almost. That shows up better on this graphic, I think. The one on your left shows you the color bar, shows you the productivity. I've converted it now to grams carbon per meter squared per day, a more traditional measurement. And it shows in the Providence River, the Upper Bay, Ohio Ledge area, Greenwich Bay, we have way more than 2 grams carbon per meter squared per day. Still pretty eutrophic with a 30% nitrogen reduction. However, here in 2013, hardly any area of the open bay is more than 1 gram carbon per meter squared per day. A huge decrease spatially, and a vast change, actually. And if you've been out in the bay, you've probably noticed that the water clarity is now really improved from the lower bay to the upper bay. There's very, very little difference. This shows the light extinction, K, versus various surveys we've made over the years. And you see that blue bar representing the upper bay decreasing slowly so that it's very close to the same as the red bar, which indicates the extinction of light in the lower bay. So it's just more proof that the water clarity is the same in the lower bay as it is in the upper bay. So I was expecting all along that this is another look at our habitat indicator of primary production. And I was expecting all along that surface production and the fixation of organic matter in the water column would be one of the drivers for the reduction of oxygen in the water column. And I had this great relationship with those black triangles there, the 6-year period of surface production versus number of days of hypoxia. I thought we had a really good relationship where the R-square is 0.93. And this is with a 30% reduction in nitrogen. And in fact, we had a very rainy summer in 2013, and those are the red dots on there, and our relationship is lousy, and there was a lot of hypoxia in 2013. It's not a very good predictor at all. And then we were rescued by 2014, which was a dry year, and we have really good oxygen content throughout the bay. There were a couple of problem areas still. Phillipsdale and the Seekonk River and west of the stations in the bay are really low and not experiencing hypoxia during this summertime period. So the driver for hypoxia in the bay, I think organic matter is important, but the real driver is whether the bay is stratified or not. Whether it's raining and there's a freshwater lens of freshwater on the top of the seawater, capping the bay so that it can't re-aerate. And in those years, in those summers, that's when we get the low oxygen events. So 2014, 2006, as I showed you earlier, and 2003, the year of the fish gill. 2014, on this plot of precipitation in centimeters versus summer days of hypoxia. And this is the sum of all the hypoxia at all the stations. That's why these numbers are large. See, 2013 is very bad, up around 250 summer days of hypoxia, versus 2014, which has just slightly over 50 days. And that's mainly in the Phillipsdale and West Greenwich area. So stratification, very important to hypoxia in the bay. So my third habitat indicator was the winter-spring diatom bloom. I have a special relationship with the winter-spring diatom bloom. It didn't do well in the warm winters between the 1980s and the 1990s because it allowed zooplankton grazers to become active. And they would graze down the biomass, and there'd be very little expression of the bloom. Something like 2005 and 2006 that you see in there. But in the early aughts, we got some cold winters, as I'm sure you remember. In 2008, we got a terrific bloom. 2010, a terrific bloom. That was the year of the flood, actually. And then 2011, we got a pretty good bloom. 2013. That was during the 30% nitrogen reduction. So the winter-spring bloom was not a bit diminished during that amount of reduction. However, with the 50% nitrogen reduction, we can see that the bloom in 2014 and 2015, and these were really cold winters. It wasn't zooplankton grazing that was retarding the bloom. I think it's nitrogen reduction. And we have further way of looking at this here. This is a plot of temperature during the bloom month versus the bloom chlorophyll in micrograms per liter. This is at the GSO dock. This is what the dean was talking about, my long-term records. And I've plotted the most recent years here in red dots, 2013, 2014, no bloom. And maybe that was okay because there's a whole bunch of cluster of points there that show that anything can happen, but not much really happens in those warmer years. But in these 2015, last winter, it was really cold. It was -1 for the month of the bloom in the bay water. And we should have had a bloom equivalent to 1977, and we didn't. So if we look at this bay-wide, the dark green line here is Greenwich Bay, still a very big nutrient problem in Greenwich Bay. And our chlorophyll levels in January, February, March reached 70, 80, 90, over 100 micrograms per liter. Whereas Conimicut Point, which certainly had equivalent levels of chlorophyll in previous years, now it barely reached 15 at 2 different times between January and March. T-Wharf, the middle of the bay, the light green line here, barely reached 5 micrograms per liter of chlorophyll. And GSO, where we would like to have about 20 micrograms per liter, barely attained 10 a couple of times during this winter-spring period. So the winter-spring bloom, one thing that's very clear, it's derived from sewage nitrogen. It's an artifact of sewage nitrogen. So perhaps it's not a bad thing that it's not so big as it used to be. Okay, the final habitat indicator I wanted to look at was fish biomass. And I got this great data set from Scott Oshevsky of the Rhode Island DEM Marine Fisheries. And I used 6 of his stations, which are underlined in yellow on that graph, going from Ohio Ledge and Warwick Light, where the bay does experience hypoxia and where you might expect some improved habitat, down to Hope Island, Fox Island, Gould Island, and East Passage, and then Bonnet Point. And I summed the fish biomass for all those locations and created that upper graph of average tow wet weight per year in kilograms. And I plotted the year prior to nitrogen reduction from 1995 to 2004 and compared it with the year 2005 to 12 with the 30% reduction. This pattern, as I try to plot this, it goes up. Fish biomass goes up with a 30% nitrogen reduction. And it goes up with statistical significance. That increase is statistically significant. And then we see a decrease at the 50% nitrogen reduction. And so far, we only have 2 years of data, and that has not been statistically significant in most of the graphs I'm going to show you. So that's the total biomass of fish caught at the 6 stations. And this is the average annual wet weight per individual in grams in the bottom graph. And before nitrogen reduction, it was about 22 grams. At the 30% nitrogen reduction, it's about 26 grams. And with the 50% reduction, it drops down to about 24 grams. And then if we look at individual fish species, and I chose benthic species first here, this is the average total wet weight per year in kilograms. In the decade prior to nutrient reduction, we can look at winter flounder. Winter flounders, you probably realize, are a basket case in the bay. They don't change from that decade to 2005 to 12, and then they drop precipitously in 2013-14 because they had a very warm year in 2012 and that was a very poor recruitment year for winter flounder. And then the skates have been abundant in the bay. In the decade before nitrogen reduction, they were still abundant in the 30% reduction, but they dropped back pretty significantly in 2013-14. Summer flounder, the same kind of pattern, increased to the 30% nitrogen reduction and then decreased with a 50% nitrogen reduction. And then the pelagic fish, and you might notice that the biomass of pelagic fish now exceeds the biomass of benthic fish in the bay. And we looked at butterfish, huge increase to the 30% nitrogen reduction, and then a big decrease at the 50% reduction. Scup are doing beautifully in the bay. They went up at the 30% nitrogen reduction and then up again at the 50% nitrogen reduction. They're doing extremely well. Atlantic herring increased with the 30% reduction and then decreased at the 50% reduction. So what's going on here? Well, I had initially hoped that the increase in fish at the 30% nitrogen reduction was an improvement of habitat. And it could be, in part. These are the stations from north to south: Ohio Ledge, Warwick Light, Hope Island, Fox Island, Bonnet Point. And we can see that the increase in that first 30% reduction in that decade is greater up at Ohio Ledge and Warwick Light than it is at the other stations. And this is a significant increase from one decade to the next. Warwick Light is significant, Hope Island is significant, and Fox Island is barely significant. Bonnet Shores is not. But the big increase in the upper bay that does experience hypoxia may indeed signify some improved habitat. But the other stations don't experience hypoxia, so I wondered what was going on. And the first I had a heart attack that the DEM changed their boat. They got a new boat in 2005, and they got a new trawl. The trawl had the same dimensions as the old trawl, but anytime things change like that, you think they might fish differently. So I compared Jeremy Colley's data set for the Fox Island station with the DEM data set for that decade prior to nutrient reduction, during the 30% reduction and the 50% reduction. They agree really well. There's still some changes between the two. I mean, the DEM boat has 6 millimeter mesh in its cod end. The GSO boat trawl has a 25 millimeter mesh in its cod end. The DEM is a 20-minute trawl. GSO is a 30-minute trawl. DEM samples once a month. GSO samples 4 times a month. So there's still a lot of differences, and for all that, they agree very well. So I'm pretty convinced it's not an artifact of the trawl or the fishing characteristics of the DEM boat. But this next graph, I think, explains why there's such a huge increase of fish during a 30% nitrogen reduction. This is the GSO Fox Island demersal invertebrate mass. DEM doesn't measure invertebrate mass. They only measure fish or commercially important species like lobster. So this is the mean total wet weight in kilograms per year, and it decreased hugely between the earlier decade and the later decade, and that decrease is due to a decrease in crabs in the bay. Cancer crabs primarily, but also lady crabs, blue crabs, spider crabs, have all, including lobster, all these decapods have decreased in the bay. That decrease is almost 84%. So much of the improvement we saw in fish biomass from prior to nutrient induction to the 30% nutrient induction could be due to the loss of crabs in the bay. And I know I'm getting way off my point here, but I know you're going to ask me too, why would crabs in the bay decrease? And the top graph there shows the DEM data for the 6 stations, and the blue line is scup going up and up, and the red line is lobster going down and down. That's the only decapod they measure. And I think that both scup eat crustaceans, about 10% of their diet according to the literature. And we have a new player on the scene here with the black sea bass, which are, I had to multiply them by 10 to get them to fit on this graph because there are not very many of them yet, but 25% of their diet is crabs. So I think some crab predators have moved back into the bay and decreased crabs in the bay. And the GSO data is shown here in the bottom graph, where you can see the scup going up and up and the cancer crabs going down and down over those 2 decades. So not all of the improvement in fish was due to a 30% nitrogen reduction. And at the 50% nitrogen reduction, it's still not as low in fish biomass as it was prior to the nutrient reduction data in general. So what are the conclusions from these observations? We've seen an average decrease in dissolved inorganic nitrogen of about 64%, an average decrease in phosphate about 57%, well within the guide, the goal of the Rhode Island DEM. And we've seen that in Providence River and the lower bay, the decrease in metabolism has been smaller, on the order of 16 or 17%, whereas in the mid-bay, much of the middle of the bay, it's decreased on the order of about 30%. And with that decrease in the production of organic matter, water clarity has improved throughout the bay. We have seen that hypoxia was not decreased during a wet summer. But we've seen that during a dry summer, it was very much decreased. It was a big improvement. And we've seen that the winter-spring bloom has been reduced by low oxygen. The 50% nitrogen reduction has definitely reduced the expression of the winter-spring bloom. And we've seen that the fish trawl biomass increased with a 30% nitrogen reduction, perhaps due to the decrease in crabs in the bay and the opening up of more habitat for fish. But we've seen that at the 50% nitrogen reduction, that fish may also be somewhat reduced. So what would Goldilocks say about the 50% nitrogen reduction? Was that just right? And so what would you say? We've seen that the decrease in primary production in the bay has increased the water clarity throughout the bay. We've seen that hypoxia has been very much improved during at least a dry summer. We've seen that fish biomass is at least as good after the 50% reduction as it was prior to any nutrient reduction. And so maybe we've got the nitrogen reduction about right. I think it's up to you to tell us. And with that, I'll answer any questions you may have. Yeah.
Yeah, Candace, have you also shown then that except for Greenwich Bay and 1 or 2 other places that the role of atmospheric deposition and and runoff, urban and agricultural runoff, are pretty insignificant here.
Yeah, the atmospheric deposition has been measured on Prudence Island. I have never actually done it, but it has been measured in earlier years, and it's measurable. It's there. It is an input to the bay, but it's not large compared to wastewater treatment facility inputs. And we don't have a lot of agricultural runoff into the bay. Most of the nutrient input to the bay is derived from urban areas and people and concentrations. So that has generally not been a problem. And with all the wastewater treatment facilities in the watersheds also instituting this treatment to reduce nitrogen in their effluent, the nutrient input to the bay is pretty low, even in runoff situations. Yeah.
With the fish and decapod data that you showed, do you feel comfortable associating the changes that you've been seeing solely with nutrient reduction, or do you think other factors like water temperature, fishing pressure, etc., might be a significant factor?
Yeah, I tried to figure out what factors were important, and the last time I gave this talk, I suggested maybe the increase in temperature in the bay was increasing fish biomass in the bay. But I really think, I hadn't realized until I looked at Jeremy's colleagues' fish data that the crabs had decreased in the bay, and that decrease was on the order of over 80%. And I know that when crabs first came into the bay, when we lost the boreal fish species because of warmer temperatures, the fish biomass increased, and we attributed the loss of crabs to predation, lack of predation, because those fish had declined to such low levels. But now I think some fish predators are back, and I think the crabs are now decreased because of predation. So I think it's crabs. It's a wild speculation, but I think it's crabs. Yeah.
I'm struck by the fact that Greenwich Bay, once again, we know is, from a water quality standpoint, a big trouble area. But I just finished reading our new book on shellfish, and Greenwich Bay remains the most productive area for production of mercenary. How do you reconcile that? And would a further reduction in nitrogen, do you think, have a negative impact then on quahog production as the most important part of the bay for?
Yeah, you have to be careful what you do and get unforeseen effects. Quahogs are really unique in that they don't mind hypoxia. It kills their predators, basically, and they can withstand it and do just fine. And I do think the high productivity of quahogs in Greenwich Bay is probably because it's got an enriched environment there. But I think it's still, I mean, they have really improved Greenwich Bay a lot, too. It's not particularly noticeable in what I've shown you here, but in fact, the nitrogen input to the Greenwich Bay is also down. And I worry about the 50% nitrogen reduction, why that might decrease secondary production in the bay. But so far, everything is okay. Yeah.
Yes. It seems on that graph that you had, though, that showed Jeremy Colley's fisheries data versus DEM.
Yep.
That his showed a steady increase in
yes, it did.
Between each
Yep.
stage.
Yep.
The DEM data showed a decrease.
Yeah, so decrease at the 50%. Wasn't shown at Fox Island down here in the lower bay. And you know, I don't have an explanation for that. I mean, as you came further down bay, the effect of the increase at the 30% nitrogen reduction got less and less. And Fox Island was barely significant to begin with. So if the DEM and the GSO fish trawl, they agree pretty well, I think, considering all the variables involved. I'm happy with it. Veronica.
So Candace, you said there's a decrease in the winter spring bloom, and I know you love the winter spring bloom.
Yes, I do.
What do you think the implications are of having a decreased winter spring bloom all the time now?
Well, I think that the boreal fish and the feed of boreal fish might be reduced, but I think boreal fish are already reduced in the bay, and I think pelagic fish are now dominating that do not depend so much on the winter spring bloom. So I think fish-wise, we're okay with the loss of the bloom because the boreal fish already are not here because of warmer temperatures. Don't know. Kim?
Do you see any evidence with the reduced blooms or production of it just not getting to the benthos?
I haven't actually looked at that, whether there's less organic matter going to the benthos or not. But some of the work by Wally Fulweiler and others that are measuring benthic fluxes and respiration has showed during that period when we had very low winter-spring blooms in the 80s and 90s that the respiratory rate of the benthos was very much reduced from the earlier data that we had gathered during a period of higher winter-spring blooms. So yes, I think the loss of the brew does change the food environment of the benthos and decreases it probably quite a bit. Yep. Angelo.
Yes, sorry, just one other thing. I think you were grouping 2013 with prior to the 50%, but I thought in the recent data we showed 2011 and 2013 were very different, and 2013 was very close to 2014, 2015.
Yeah, 2013, the nutrient reduction that I could measure from our standing stock concentrations was achieved during the summer of 2013. It was not during the winter, it was still high that winter, but during the summertime the nutrient reductions were at 50% level.
Okay.
But not 2012, it was still high in that summertime period. Yeah. Tom?
Professor, you indicated that nutrient levels are still pretty high in Greenwich Bay. Do you know what the DIN number is, average seasonal DIN number is in that region? Because the wastewater plant's already reduced there.
Yes, they have. They've done a pretty good job. I think one of my early graphics here, so I can go back up there.
I know there's still talk about groundwater and failing septic systems in that region.
I think it is a very large groundwater input of DIN into the Greenwich Bay area. So this is metabolism. Let's see, TIN. This is DIN. Here's Greenwich Bay. You know, they've had it, it's up and down, but it's some years it's pretty good, and other years it's not. The 2006 to 10, the 5-year period, the subsequent years are pretty low, but 2014 hops up again there. So it's been variable.
Okay.
I can answer that a little bit.
Yeah.
Greenwich Bay, the Watershed Watch is a set of volunteers who have been monitoring 5 of the freshwater tributaries that had some of them had a heavy septic system presence. And we've been tracking that, so some work we did in 2000, and the nitrogen concentrations are going down in those freshwater tributaries, which is probably a good survey for groundwater. They are still pretty high along that northern shoreline, looking at the Tuscatucket area.
Mm-hmm.
So we do track, and their data actually is online as well. So that there is still a good input from a legacy. There's one small sub-watershed that was high density residential, primarily on septic systems, and we can kind of trace, they actually all tied in around 2001, 2002, a very high percentage. It's a small enough watershed you can kind of keep track of that. Yep. And even that one, you can see it tailing off, but the concentrations in that stream are still, I don't want to put a number on it, but a couple milligrams per liter at least. Yeah. You know, so they're still pretty high.
Candace, some of the data on irradiance was striking, with light penetration.
Yep.
We should be seeing huge increases in benthic diatom productivity and recovery of eelgrass beds. Is anybody noticing that?
I don't know about, I know that eelgrass beds in the lower bay have been recovering, the ones over in the East Passage. I don't know how the ones that Save the Bay planted in, say, the mid-bay region are doing. I have no idea. And I don't know about benthic diatoms, whether there's more of a flora out there on the benthos or not in the shallower areas of the bay. I would suspect you're correct. One of our data gaps, I fear. Yes?
Yeah, so thinking back to the Scott Mason's thing, make sure historical, make sure.
Yeah.
With the reduction, the 50% reduction, can you, I'm not a reduction person, can you place any time, like a timeline of where that would be kind of historically?
which, I don't know.
You know, I haven't tried to do that. That would be a very worthwhile thing to do. I suspect we're getting close to historical levels of nitrogen in the bay at this point, and phosphorus. I mean, 50% reduction is quite a big jump. But I don't know if Verrazano could see his toes yet or not.
What history are we talking about?
Good question.
So Candace, is this study just getting underway? Are you continuing?
No, it's wrapping up. Yeah, it'll finish as of this June, actually. Well, actually, I guess it's August, but we have to get a final report in June here. Yeah.
Great. Well, I'd like to thank Candace for a very interesting and inspiring talk about the research that she's done for a very long time here and the contribution